So if you start cursing now, it's recorded forever as opposed to just, you know, what the people in the webinar are seeing. I hope nothing goes viral. All right, welcome back. This is day one, part not that. Okay, that's fine. I thought I typed in something different. We are now day one, part 4. We're going to do a metagenomic experimental design with a little bit of a hands-on twist at the end. Okay, all right. So hopefully this section doesn't take too long. I can start to yammer on about metagenomics. This is something that I've been working on for a pretty long time. I'm gonna turn that off. So I like to just share with everybody as much as I can about it, so it can be a little bit much. So if I'm rambling on, somebody give me a wrap it up signal. There we go. And so I just want to start off by defining a couple terms that I'm going to use. One being metagenomics, which I've said a number of times already today, which is the random sampling and sequencing of genomic material from the environment. That's all it means right now. What you're actually targeting, how you're getting the DNA, all that stuff is some stuff that we're going to cover right now. But that's the term. That's what I mean when I say the word. And then another term that we've used a couple times today, but I haven't defined, is contigs. And so contigs represent a contiguous stretch of DNA that are composed of smaller fragments via some form of assembly. And so that's what you do when you get your raw reads, which start small, and you want to make them into larger data. The large data are called contigs. All right, so let's do a little bit of a history, a little background on this. So the first metagenomes. So these were done in the early 2000s, and what they required you to do was to take your sample, normally from the marine environment is our example that right here, but in most environments you'd have to do something similar to this, where you take your raw sample, concentrate your cells down into some capacity, and then what you would do in this really terrible method that was not high throughput and took a lot of time and you didn't have to Sanger sequence everything, you would extract the DNA and put the DNA into some type of carrier molecule. And so these molecules could range in size from plasmids, which would be about 10 kb, to phosmids to BACs. And so BACs are bacterial artificial chromosomes, and they can be very large, and you could put lots of DNA into a BAC, but at some point you still have to come back and sequence it. And so the sequencing at this stage was all Sanger sequencing. And so what you would do is you'd get your organism, organisms, you just extract their DNA, you put them into a carrier molecule. That carrier molecule would then go into E. coli. And so you'd run into this problem where if the molecule that you put in, the DNA that you had some protein that was expressed that killed the E. coli, you'd never see it. It would be lost from the world forever, and you would never know it was there. And you'd also run into this problem that, the sequence would be too long that you couldn't actually get to the sequence by itself. So you'd have to do some methodology of screening. You could do Western blots and things of that nature, where you could actually look for the sequence that you're looking for, find out which cells have it, grow them up, extract that portion of carrier molecule, either with a plasmid extraction or a BAC extraction, and then painfully work your way across the sequence using Sanger sequencing, and getting little tiny fragments that you can stitch together and to make long fragments. This was extremely powerful. Like, this was how the GOS metagenomic data set was generated. And we discovered that there were proteorhodopsins in the ocean. So that was pretty cool, but it took a long time, and it wasn't the most effective way of doing it, especially now that we have these really high throughput methodologies. So that's what we got here. We had the Sanger sequencing based off of GOS, and we have discovery of proteorhodopsins. Awesome. We also could do this in the early 2000s. You could do it in very simplified communities. You could take these Sanger fragments, you could stitch them back together, and you could reconstruct genomes of microorganisms. And this is where metagenomics, when people were starting this in the early 2000s, this was the claim that we were all making, is that we could use metagenomics to reconstruct environmental microbial genomes, and we could get past that limitation of culturing organisms to be able to study them. And so this was the first one from an iron mine drainage. And so these were two organismal genomes. It was a very simplified community. You could pull these genomes out and say something about it. In going back there multiple times, people have realized that these two very large, clear genomes actually represent the most abundant organisms, but they were actually missing still some of the more rare organisms that were strains of, these the organisms that they originally identified. So we had this old methodology that was very useful, but was fairly low throughput and really could only handle certain types of data. So then we entered into the new phase of sequencing that we're in, the next generation of sequencing, starting with the 454 sequencer and moving forward from there to the more modern versions of it, where you get more sequence for less money. And so at the end of the day, despite the success of things like the Global Ocean Survey, the first sample that they got back from the Sargasso Sea, even with large fragments of DNA, because their coverage was so low, there was a very small amount of DNA that actually went into large contigs, right? So contigs greater than 10 kb, things that you could take a look at. So 10 kb is about 10 genes. So there was only a small percentage of the DNA going into these long fragments. And so everybody was like, well, this is a coverage problem. We're going to do coverage. We're just going to do next generation sequencing with the real small fragments, and that's going to solve all the problems because we're going to have much higher coverage. And then we ran into this problem that when you get to much higher coverage, all the tools that we had designed for Sanger sequencing don't work anymore. You have to completely revamp some of the pipelines required to do assembly. And so you could generate still lots of data. And there are plenty of studies from the late 2000s where people went in and did lots of 454 sequencing to generate as much data as they could. And at the time, it just really didn't have a result that people could get back from, where the goal was reconstruct things that look like microbial genomes. But we could take that data and look at what we saw there just based off of the fragments that we're getting back. And so this is moving towards the end of the decade in the 2000s. Looking at some sediment samples in the Peru margin, where we could actually look at the taxonomy of the reads that were assigned and do some comparisons about what the environmental organisms look like. I would imagine if you went back and redid this, it would not look the same. But at the time, this was a PNAS paper where we could actually say, look, we can take these large data sets of metagenomic sequences, and we could say something about who are in the community and how they change, in this particular case, over depth. So, if we were generating small reads and things weren't working, we needed to do stuff on that, about that. And so it took some time, but we developed algorithms, and algorithms have developed that allow the small read data to be incorporated more effectively. And that allowed us to then also do, once we had better assemblers, we ended up designing algorithms that would allow us to take the new assembled fragments and contigs and start to figure out ways of clustering them. And so these were ways of binning sequences that were related to each other. So we ended up getting to this point where, okay, so we weren't going to reconstruct all the time one single molecule of bacterial DNA. But if we have long enough sequence fragments with our newly designed assemblers, and we have the right algorithms to look at and compare each of the individual fragments that we're making, maybe we can come up with ways of finding fragments that belong together, grouping them together, and then looking at them and treating them as a representative of a fragment of a microbial genome. And so we can use various features to do that, like tetranucleotide frequencies, %GC, amino acid usage. So these are all ones that people have tried. And then really what has become much more of a gold standard as a way of identifying contigs that belong together is coverage. Coverage over multiple samples helps you identify contigs that belong together. And we're going to go all through that tomorrow in our binning methodology and how it's going to act and how it actually works. But what essentially you do is that organisms that are in a sample who have DNA that's in one long piece, if you have fragments of that DNA and compare it to multiple different metagenomic samples, those contigs will have similar levels of coverage based off of the abundance of that organism in the sample. So if sample 1, it occurs a whole bunch, in sample 2, it occurs very little, all the contigs that belong to that same organism should have very similar amounts of coverage in a particular sample. So you're able to identify the fragments that belong together and pull them out. This is a whole session tomorrow. It's like an hour and a half. We're going to talk about it until you're sick and tired of it, but just to give you an idea. And so what happens when you do that, right? So the first couple of papers were looking at just tetranucleotide frequencies and GC content and amino acid usage. And you get, at the beginning of this decade, so 6 years ago now, so only 6 years ago, I want to emphasize that, we were able to take a metagenomic data set and reconstruct an environmental genome of an organism from the marine environment. So this was the first genome for the group 2 Euryarchaea. And you can see it's this arrow points to it. It's this, in this very visual map, you can see it's this area that's connected all together. And so you can see it was just one long piece, and they were like, oh, it's one long piece. And they pulled it out and we had a genome. It was great. Another couple of years went by because that worked really well for their data, but not a lot of other people's. But by 2015, you have these papers that were starting to be pushed out from Jill Banfield's lab group and other groups associated with her lab at Berkeley, where they were starting to able to generate more and more genomes. And so this is one of the first ones that looked at the CPR in 2015 and identified the candidate phyla radiation. And you can start seeing that we got like a couple of complete genomes here in a very small number, 2 and 3. But then we got lots of these draft genomes that were partially complete. And we could ask how complete they were, and we could start getting these organisms together. And then we could move forward a little bit in time for the same year, and we can start asking, okay, so what are these organisms and what are their functions, and how does that work in the environment? And so we have these types of maps that we can look at where we have inputs going in through organisms and then outputs on the other side, and how these functions might be relating to each other. We can keep moving forward. And so now we're at this phase where, so this is a paper that we put out this year, where we were able to take the really large Tara Oceans data set and do a binning method based off of that. And we were able to generate over 2,000 genomes from the data set. So this is just a phylogenetic tree of that. And so we've changed. So if 2012, that year we got 1 genome from a metagenome. And then in 2015, we were getting a couple hundred a year. We're now in this phase where if you have the right genome or metagenome data sets with a large enough size and the right methodology, you can be generating thousands of genomes a year. And we see that. So last year we saw a data set that was released that had 8,000 genomes. We released a data set that had 2,600 genomes in it. There's a couple data sets in the pipeline that have about 4,300 that are coming from soil environments and other complex systems like that. There's been more coming from deep terrestrial systems with slightly smaller amounts. So we've gone from this era where metagenomics was this promise that we were all going to get something out of it and we could start getting environmental microbial genomes from it. And now, almost 20 years later, it's really starting to deliver. And we're really able to start really pushing out some really large numbers, which means that we're finding new things. So this is another paper from this year where we're able to identify for the first time organisms belonging to the alpha proteobacteria that combine type 2 photochemical reaction centers and the potential for carbon fixation, which had not been seen in the ocean before. And we see that, so new phyla have been discovered and subgroups within that phyla, things that have never been seen before, we're able to do for the first time. And so if that's your goal and that's what you want to do, we want to think about what types of protocols that we might need to think about before we start taking our samples to make sure that we can get to the results that we want. And so the first thing that we'll talk about a little bit, and so these are the 4 things that we will talk about: high quality DNA, size inserts, amplification, and depth of sequencing. All right, so high quality DNA. So this is really crucial, and especially because a lot of you are here from C-DEBI, you know this, that low biomass systems are really hard to do this with. And so being cautious and practicing any technique that you're going to use in terms of extracting DNA to get the high quality output that you want is going to be really important, especially for low biomass samples. Also, we're dealing with people in this room who are dealing with the environment. So humics are going to be really important and will impede lots of the downstream steps for generating libraries that you want for the sequencing. Metals are a similar problem. So we need to be thinking about these things and working on procedures that help to clean and extract high quality DNA. And so depending on what we're going to talk about size and insert in a second, depending on what your goal is going forward, high molecular weight DNA has a potential being more useful than low molecular weight DNA. And so this includes thinking about ways of trying to prevent shearing of your DNA as you go through the extraction process, which can be hard, right? So if you're working on a sample that has really high humics and you need to, that's probably a bad example, but if you're working on a sample that you need to really vigorously shake and lyse your cells, right? That's gonna have some impact on your quality of DNA that's coming out of it. But at the same time, you can't gently rock it to get the extraction that you need to get that high molecular weight DNA. So you can, there's some trade-offs that you'll have to get depending on what the type of samples you're working with. And so high molecular weight DNA works or is important because of this idea of insert size. And so when you're working with metagenomics and with the Illumina sequencing in particular, and we're talking about paired end sequencings, you can decide how big of a fragment of DNA you put in to get primed and sequenced in a sequencer. And so in the Amplicon example, we were talking a lot about having an insert size of less than 300 base pairs, so that way you could sequence from both sides in and get a complete overlap of your sequence, and then be able to say something about how good of a sequence you have, the quality of it in that sense. In metagenomics, you can actually be looking at it in a completely different way. So when you get to an assembly step, which we'll talk about tomorrow, the larger the insert size, the better you are at bridging areas that have areas of high repeats or low information. And so the idea behind this is that if you have an area that's just a repeat, let's just say it's A's and T's over and over again for 150 base pairs. If you have reads, or if you have an insert length that is less than 150 base pairs, right? So you're just looking at a small fragment of DNA, the assemblers won't be able to resolve that information, because every read that exists every read that comes out of that region will exist within it. And so if it's 150 base pairs long and all of your reads are 75 base pairs, you'll never know once you put in a 75 base pair fragment where it belongs on your scale of that repeat region. If you have a longer insert size, so that way that the reads coming in from one end and the reads coming in from the other end start to overlap more on the outside part of the, so that way if you have something like 75 base pairs and a lot of it overlaps on the end and a lot of it overlaps on the other end, you can start building up and through and realize that there's a repeat region there. And you might not ever completely resolve it, but it allows you to start tackling that question. And so larger insert sizes can be really useful in your metagenomics. So you can go, you see people commonly using 300 base pairs, 800 base pairs. These size ranges can be really impactful in the output that you're getting. So amplification is sometimes the thing that has to happen, especially if you're working in low biomass. So right now, the kind of really acceptable way of doing it is through random linear amplification. And so there are lots of kits that can do this. One of the example would be the Nextera family of kits. And so what these kits do is they take your size fractionated DNA and they will randomly prime the ends of it using blunt end priming. And then they will amplify up your DNA using a PCR step. And so you actually get a fairly good, if you're doing multiple samples, you want to make sure you're doing it in a similar method so that you have the same biases, but a fairly good a general amplification of these fragments of DNA in a way that doesn't overly bias your samples and it doesn't affect your ability downstream to assemble and make bins. Ideally, you want to get a sample that you can extract enough DNA from. With the time of the writing of the PowerPoint, Illumina still was very much recommending 1 microgram of DNA. It's been much, we can drop that now quite a bit. 500 nanograms could probably be sufficient at this point, if not a little bit lower as your input starting amount of DNA. And then the big no-no, and this might not need to be said as much as it needed to be said 3 years ago, which is MDA or rolling circle amplification will just not make it possible for you to do the next step that you want to do, which is assembly and binning. And that's because MDA will skew your results. It selectively generates certain regions of the genome. And once it finds a region and starts amplifying it, it selectively amplifies that region it's already amplified. So if your goal is to use coverage to figure out which contigs belong together, if you have one section of your DNA that's been highly over amplified compared to a different part, it's going to look like two different organisms. And so we end up with that problem. This generally is not something that people are even doing. If you're looking at an older paper and you're like, wow, this paper was really successful and it comes from 2014 and they used MDA, just understand that that was something that was very common then, but is not common now anymore. MDA is used for SAGs, so single amplified genomes, and that is okay. But that's generally because they're using either a single cell or one cell within a population and amplifying the same DNA, and they'll normally do multiple reactions and things like that to balance it out. But they don't have to go through as much of the binning process as we do if we're doing straight metagenomics. And then this goes off the edge. So the last thing to think about is what you actually want to do with your metagenomics. So I'm coming at this assuming that people are ideally hoping to maybe get a genome or two that they can work with and do some work with that. But there are lots of other ways to use metagenomics that are equally as powerful. And so I break this down into this idea, and we'll talk a little bit more about it tomorrow, about this idea of low depth sequencing and high depth sequencing. And so a low depth sequencing might be a MiSeq run where you're only generating maybe 200 million reads at the most, a portion of a HiSeq run or a HiSeq lane where you have multiple samples paired together in some multiplex kind of situation. And this is really good if you have some geochemical data, right? So you're a geochemist and you see some really cool changes in your system over time. You know, I'd really like to think this is the bacteria. How can I do this? So you can do a low sequencing run and look specifically for the types of genes that may be working through that process. And once you found them, you have the evidence that you need that biology is playing this role in your geochemical process that you think is happening, which is great. You can do things like find genes of interest, right? So you can go to a metagenomic sample, you can say, I wonder what genes are here. Let's see which one we can figure out. We can just treat it as an entity for which we can look at genes of interest. We can do things like pull out full-length 16S rRNA from a fairly low depth, right? So that's going to give you full-length RNA sequences with the ability to look at some phylogeny to go along with that. And then we can also look at just looking at the reads themselves or of genes, taxonomy, phylogeny. You're really interested in the NIF-H gene. There's an environment that you think that you can go in there and find it instead of running an amplicon sequencing run on it because you want to look at it from a non-primer-based methodology. You could go in there. You could find that gene in your output. You can pull it out, build taxonomy off of that, and just ignore the fact that you don't know what organism it's coming from. On the reverse side, high-depth metagenomics, so either multiple MiSeq runs or large fractions or whole lanes of HiSeq, are really good for genomic reconstructions. This allows you to do binning, and it allows you to look at variable coverage between samples. So those were the 4 points I was going to point out to you right now. This always generates a lot of questions about, I have a sample that does this, I have a sample that does that. I'm happy to talk about it whenever you want, whenever we have time. But in the effort to get us done by 5 o'clock, I'm just going to move on to this next part, which is the hands-on portion. But I will take I have a sample that does this questions here right now so that we can keep it together. No. Great. Okay. So this is what we're going to do now. So there's lots of ways of doing this. And as I've said a couple times as a caveat, so this was again designed 3 years ago. I've found, so it still works very nicely in the virtual machine because the virtual machine is a pristine, perfect environment from 3 years ago. But we've even had this issue on our server where the steps that I'm going to take you through now have gone through a couple iterations. And we've actually found that the easiest way to run this is actually through a Docker image. And so Docker is very similar to this VirtualBox virtual machine situation, where it actually will create a sub environment for you to work in, so you don't have to do any installations, and it keeps it nice and pristine and perfect so that you can keep running your data through a computational system that doesn't have the impact of all these other things. And so if you see this and you're like, wow, that was really interesting, I want to do this for real, let me know and I can send you information about where to find this Docker image and how to work with it. And it will still use very similar steps to what we're going through here, but it will be just an easier way than being like, one of the programs that we're going to use is called Emerge. It's not the friendliest install in the world. So if we can make it easier, then more people will be able to use it. So we're going to be taking quality controlled metagenomic reads. So that's something we did before. We're going to extract specifically fragments from these metagenomes that have the 16S gene in it. So the gene itself is actually 1,500 base pairs. We have these 100 base pair fragments. We're going to identify individual fragments that are part of the longer read, and then we'll assemble those fragments to generate long reads all by themselves, or long 16S. Okay, so we can do this right now. So to give you an idea of how much data this is, so this is 1/14 of a paired end Illumina data run. This was one lane, so this is 1/14 of a lane that we're going to do this with. So here's an example. What are we doing? Let's go. Q. We're going to go into go back out to EcoGeo. We're going to jump into the Emerge tutorial. And so you're going to see here we have 2 compressed FASTQ files, and then we have a pre-computed area directory. We'll talk about that in just a second. It's going to keep some secrets in there for us for right now. And so what we want to do is what we did before, which was use cut adapt to remove sequences. Yeah. And so I'm going to use a shortcut here. I'm actually going to. Is this okay if I do it like this way? If I'm going to do a copy and paste, I'll make it, keep it big, and then I'll copy, and then I'll paste, and hopefully everybody can still see it. So it's going to take a second to run. Okay. I was starting to get a little paranoid right there. It's gonna be like, oh no, it's never gonna finish. Okay, so once you have read 1 completed, you can do this in a fairly simple fashion. So we're gonna run the exact same thing, but with read 2. So all you need to do is hit up, and there are 3 R1s that you need to change to an R2. So how about the results of that Croatia game, huh? Crazy. No, nobody? I just want to really date the YouTube video part of this. How about that 2018 World Cup? Everybody watching this in the future. Let's just imagine what this would be like if this was real data and not 1/14 of the data. All right. So I'm going to wait a second before we move on to the next step, but let me know when you're there.
When you're running steps like this, if you have like a set that you usually do and you know works, would you make it into like an executable file?
Yeah, that's exactly what I would do, especially if you're running a lot of them, right? And that's exactly what it is. So we received this data as 14 separate subfiles. And so I just set it up to run each one individually, and I walked away from the computer. So does most of the people get that? Yeah, we got some head nods. Things are running, have finished running. So again, I'm gonna do a, for the sake of time, copy here. So this is going to run Trimmematic. So this version of quality control that we're doing here, if you're on YouTube and looked at the quality control segment that we did before, so this is combining both the cut adapt step and the Trimmematic step, breaking them out into separate aspects of it. Unable to access that file. Why? Cool, thanks guys. Let's see what we can figure out. All right, so I'm going to try and do it like this, where I've typed it out so that it works. And I'm going to just copy from PE onwards. Let's see how this works. Ah. That's a great question. Let's see. There's probably a typo.
The error that you can't access it?
Yeah.
Well, it worked the second time, didn't it?
Yes, but I skipped this part. So I'm just going to hit paste and see what this looks like different. Ah. Programs. The original version of this didn't have an R there.
Oh, it's programs.
It said programs in the copy and paste version. But if you put an R after the G,
Spell it.
yeah. Yeah.
Okay.
I mean, that's so weird that spelling matters. Didn't somebody say at the beginning of all of this that like it doesn't autocorrect and that you have, it's not like your phone and you have to make sure you type out everything accurately?
Well, it's not giving me an error.
Great.
I mean, it'd be fine to misspell it. You just have to always.
Yes, that's true too.
It'll always, always be
Programs. Okay, so this output that we get after you've fixed my typo and made it work a little better, it should have about 86% of the sequences are being saved. So this was a much better sequencing run than the one that we were looking at the beginning of the day for the quality control metric, right? So remember, with that one, we threw out almost 40% of our data. This one, we're only throwing out about 15%. So this says to me that we've probably got a slightly better sequencing run from this data set than we actually did from the one that was used earlier today. Do most people have that?
Mine's still running.
Still running. Okay, well, we can wait for a second. So if we look at the output while it's still running, so now we have still a whole bunch of compressed files. That's what the .gz means. It's also indicated by the bright red. So these files are compressed to save space and not to generate, take up the full footprint that they are, to show that you can do these commands using compressed files. But what we need to do is, if we want to work with the rest of the programs that are listed here, we actually have to uncompress all the ones that we just made really nice and clean. And so these are the R1 and R2 underscore paired .fastq.gz. And so we can use the gzip command, -d, which is gzip/d, or the flag is decompress here. And do R1 underscore paired. We can hit enter, and that'll take a second. And then we can hit up, and we can change it to a 2. And then so the next program that we need to use won't accept 2 paired sequences, right? It won't accept one file with the first one being the first number. It won't accept the second file that way. So what we need to do is what Kristen had mentioned before, is we need to do something called interleaving. And so what interleaving does is it takes our 2 files where the first entry of each file match each other, and it makes one output where the first 2 lines now are paired reads. So read 1 forward and read 1 reverse are now going to be right after each other, and it's going to go down the whole way and do that. And so we're going to use that using a command called FQ to FA, which is just FastQ to FA, and we're going to merge them. So we're going to use the merge flag. So it's going to be --merge --filter. Then we can take R1 paired, R2 paired, and then our output is going to be, I'm going to name it NP 2014 ctd.merged.fasta. So this is just interleaving. It's also going to convert the FastQ files to a FASTA file. So if we look here, so we now have our uncompressed files here, just like that. And then we have our merge FASTA file here. So the difference between these 2 files is that these are not interleaved, they now are. And these in the R1s, R2s were FastQs, and now we're working with a FASTA. All right. So now we have to look for 16S rRNA fragments from our merged FASTA file. And so we're going to do that using a program called MetaRNA. Again, this is on your install list, so you can find it there. What we're going to do on this slide is we're going to detect the 16S fragments, and that's going to take this nice long command that happens here. We're going to actually be using, so this is a Python script that then looks at and uses an HMM model, and we'll talk about HMMs tomorrow, to identify fragments that belong to the 16S. And so we're just telling it very specifically that we're going to be looking for the small subunit, and that we're using this really nice high, or low in this case, E value to find really only things that look exactly like 16S and not things that maybe could be 16S. So in the current version of MetaRNA, the rest of this slide actually gets done for you as part of the program. It doesn't in the version that we're using on the virtual machine. So there's a script that is part of the programs that we have that will actually convert the output from MetaRNA and then take our input FASTQ files and generate a 16S fragment FASTQ file that we can then use for the assembly step. So I'm gonna copy and paste and see how this works. Here, programs is spelled correctly. And so what this does here, which part did I not like? There's a new line somewhere that it doesn't like. Like right there. There we go. So it should be, so what we're doing first is we're calling the program, which is located in the Bioinfs program section. So it's home, C-DEBI, Bioinf programs, the name of the program, which in this case is RNA underscore HMM3, and then the program itself, RNA underscore HMM3.py. Our input, which is our FASTA file, the NP14 CTD merge file, -L, which is for their library input. So we're telling them where to look for the HMM libraries that are going to be used to make the search, which is in the same directory as the program, but in a slightly different subdirectory. So instead of writing the program name, we're going to write the directory that it's looking for. We're going to output a table that is going to give us our predicted RNAs. We're looking for the small subunit, and we're looking at it for a fairly high level of specificity and threshold cutoff. So if we hit enter, it's going to hate me. Say this parentheses to call print. Oh boy, that's not great. All right, let's try typing it out. I'm just going to hit enter here and see if we get the same. Yeah, this is an error. So, Kristen, I have to ask you this really quickly. How do we get out of the chime? Just type exit? No.
Take it over to the virtual box.
That's not. Now we're out of chime. So the problem is that chime, if you still have the parentheses chime source ID, the problem is that that's running a different version of Python than the Python that the program is running. So we're gonna exit out of that for a second.
I still got an error, but I wasn't doing that.
Great.
Yeah,
So maybe we're even more.
I got.
Okay, so we're gonna try one more time here, and we'll come around the room to make sure everybody's on the same page. Yes. Ah, now it's running. So I'll come by. Okay, and so now the only issue with this one, so we don't need, oh, so you're working on the next step.
Isn't that the step one?
You're doing, so this one worked for you. That ran for you the first time, right?
That one ran.
Yeah, so I haven't gotten to that step yet.
Oh, you haven't?
Yeah.
Oh, okay, sorry.
You didn't even have permission.
Well, just as a heads up, there's a problem with that next
Yes,
step.
great. Now looks awesome. So this is saying that. Yeah. Yes.
Bioinf tools.
Right, because again, that's
Yeah.
where I was before, and then I closed it.
Right. So you got that running.
Or bio programs. Yeah.
Yeah, that looks like that worked. And you're on to the next step now, right? Yeah, okay.
It's frozen in 20.
Cool. All right. Well, I'm gonna walk away from that.
Yeah, that's not a
Yeah.
fixable problem.
I already did that.
Okay. Now it looks like it's running. How's yours going? Well, right now you don't have any of
Oh, me too. The second we wrote about.
The second,
Yeah,
yeah, so
it
it didn't
says
say there's
no
no,
module
well,
name.
named bio, it's okay, that's fine.
But the funny thing about it is I don't see.
You do the opposite of source activate, you source deactivate.
Oh, that makes so much sense.
Your version.
How's everybody doing? Okay. So here, so this is the error right now.
Yeah, it's something, there's something wrong with the script.
This is
From bio imports.
copy it.
Something in bio module.
You need this, right?
directory. So you need to go to home.
Yes. All right. How's this table doing? All right? Yeah. Yeah.
It's also a document file, so you're trying to execute it.
Oh, man.
So, oh, you didn't just see yeah. You should have. Okay, now there's no file. There is no
Oh, so now it's saying no.
There's no program and there's no other. Let's try to import to see if I have a
Ah. Okay. Yeah.
No, I think, well, we looked it up. There is no module named bio.
Oh.
It's calling it for meta RNA. And it's not executing.
Okay. Everybody's got that part running. So now we're running this next problem where everybody's saying that this isn't working for them, correct? Okay. So let's see. Okay. Dash R P. Okay. Great. Bio.
There is no bio module.
In the virtual machine, huh?
Yeah.
Or in the, yeah, you need to go to
In the bio
RNA fast cube slide or something.
Okay. That's a new development for me. Module name bio. Dislike. This is not how that's supposed to work. Okay. So that's a fascinating new development. I'm going to blame the new Chime download into the virtual machine on this. I can't guarantee that that's the case, but that's what we're going to do. We're going to point our fingers over that way and hope for the best. I can get into this in a little bit and we'll figure out the solution. As I said, the new version of meta-RNA doesn't require this step anymore. So that would be very beneficial at this point. But we should be able to finish up with this as we go forward, right? And so because the next step we actually end up having to just run independently from here, and I can fix this, make sure that it's not hanging out here and pre-computed. It's not. Okay, great. Okay, so the next step would be, so after everybody, we got the meta-RNA to run. I think everybody in the room actually ended up getting that to work properly. what happens is meta-RNA will generate an output where we then have taken our reads and will only give us back fragments of those reads that are that belong to the 16S RNA fragment. So if you had a read that's 100 base pairs or 75 base pairs in this example, and only half of that is 16S, it will trim it and give you back just the 16S fragment of it. And so we end up with both our forward read, our R1s and our R2s, generating a FASTQ output. And so we can combine those, right? And so if we had these files, which we don't, and that's okay, we would use cat, and we can use the wildcard flag to just find everything that says 16s.fastq and output it as a new file. So in this case, it's called 001 because it's that 1/14th of the data set that we're using for. That's why I named it. So at that point, we have a truncated FASTQ file with the 16S fragments. And then what we can do is we can run a program called emerge. And emerge uses a silver reference database. In this case, in this example, we're using an older version, 1.1.1. Right now they're on 1.32. There's a whole method and steps on the emerge page to tell you how to update that database so that you can use the most current version, most up-to-date version. And what we do is we pass our fragments, our identified fragments as a whole into emerge, which is what this is right here. We then are using the Silva reference database as a way of kind of marking where those fragments should go in a 16S sequence. We tell some information about the data of the sequences that we're giving them, the maximum length, the input distance between the two fragments, some extra information about what type of quality scores we're using. And what we get on the output is we get, so if we end up, so if we had the ability to, we could count how many sequences we're putting in here. I believe it's only a couple thousand, 600 or whatever. So if we actually did this with just the 1/14th, we actually don't have enough data to generate a full length data set. But so what I did was I went back in time and I actually pulled the full data together for you and ran it using emerge. So what emerge did was took 29,823 16S fragments identified from the full data set and ran them together. And so it goes through this whole process. And in our pre-computed folder, after the 40 iterations that emerge runs, we have an output, and that output we can actually rename and have a final 16S sequence to work with. And so we can do something like run the script emerge .rename _FASTA like that. And then if we do a / pre-computed, we're saying, look in the pre-computed folder, there's a folder in there called iter.40. And then if we just run an output to a name like np 2014 ctd.final 16s.fasta and hit enter, what it's done is it's taken the output that was in that iter40 directory and has automatically renamed it as a new FASTA file, which has our data in it. And so emerge works in this iterative process, right? So you can actually set how many times it goes through it. 40 is the default. And what you'll see as it's running is you'll get these flashes of iteration 1, and it'll tell you how many reads have been incorporated into a 16S sequence and how many haven't, and it'll run it through iteration 2. And so you can actually go back through if you wanted to do something like this and look at what the 16S sequences look like at each of those iterations. But once you get to the final iteration 40, you can go in, you can pull out that final number or that final file and have your full length sequences. Yeah.
Can you explain exactly what emerge is doing? Is it taking fragments,
Yep,
stitching them together to create full length fragments, and then the final output are only the completed ones, or no?
So what it does is it takes your fragments and it uses the reference database that you give it as a way of scaffolding where they're supposed to belong. And then if they overlap, they stitch it together. And if it doesn't overlap, but it has a high confidence that this is the 16S fragment that it's most similar to, it can actually build you a full length fragment that has gaps in it. And so the more rare and the less The less an organism occurs, the more likely it is to have a gap, but it will only generate a full-length sequence at the end of it. So if your organism is there at such a low abundance that it can't figure it out, or it's so far removed from the database that you give it that it can't align it properly, then it won't be included in your final data set. But it does give you an almost approximation at that point.
So if you're working in an environment where you have a lot of novel lineages,
Mhm.
then it's not likely to produce an output with those full-length 16S.
It can, but it will be more, it gets a little bit more spotty. Yeah. Okay, so we have a 16S product despite the hiccups along the way. And so if we take and do LS, we can see that it's here, this MP 2014 CTD, final 16S. We can look at what that file looks like with our ls command. And so this is going to give us an output, which I think I covered. No, I don't cover in the next slide. And so what this does here, so this is our output. And so this is, here in the corner, this first number that we see is its ID generated by a merge, given by a merge. There's a pipe. This value right here tells you first the sequence it's most similar to in the Silva database, and then the length of the sequence that you have right here. So this is covering from 1 to the full length 1494. This is the length of the sequence that we're looking at. And then there are two values here. One, which gives you a relative abundance of the fragments that you gave a merge, right? It will tell you how many of those fragments have recruited into this full length sequence and gives you an estimate of what its relative abundance was in that sample. And then, so that's what this one does. In this version where it gets normalized, so some of the sequences have different lengths. So the bacteria have a 1400 base pair general length size. Archaea have a 900 base pair length size. And so it normalizes based off of the length of the gene that you're looking at, right? So a smaller gene will recruit less reads, but it doesn't mean it's less abundant than something that has a longer read. So we can actually scroll through this and we can start seeing that there's other ones. So the next one occupies 19% of the sample. And if we keep going down, so now it's 4%. And if we go to the bottom, so I'm going to just fast forward to the bottom here. We start getting into the 0.2% range, and we can keep going lower. And I'll give you an example of ones that start to have gaps in them. So yeah, so here is one up here. So you can start seeing that there are these large stretches of ends. So this was one that it could align pretty confidently. You get these fragments that all belong to the closest relative in the database, but then it couldn't complete it. So it actually would fill it in with ends for you. And so this is an organism that's about 0.1% of the community. And so we can do what we were doing before, just as a reminder. We can do grep, open quotes, caret, close quotes, NP, CTD, final. And we can word count, Gesundheit. And we can see that this generated from our full data set about 303 full-length 16S sequences. And so we're not going to go too deep into this, but if we want to, we could go and we can figure out what these who these sequences belong to using some type of taxonomy. And so we can do something like we can use BLAST against NCBI. And so if we were to do that with our top sequence, we'd get our output that looks like this and says that it belongs to an uncultured clone. It's a full-length 16S sequence. It's 99.9% identical from some other clone that's existed before. And then in this example here, I do a walkthrough of if you wanted, you could take your output that you're generating and use something like Mother to generate a taxonomy, a much faster, more robust, high throughput version of taxonomy, where you use align seeks and classify seeks within Mother to generate an output that then you can search through. And so if you were to go into pre-computed, because that takes some time to do, so you can CD pre-computed, there's a taxonomy file in there, so we can look at that. And that gives you a similar output to what we saw from CHIME 2, where our number one sequence here is most related to a gamma proteobacteria and the Ultramonas genus. Yeah. And then, you know, we can go through to the third most abundant is archaea from the marine group 1, et cetera, et cetera, et cetera. So that is the last slide. It's very interesting that that bio didn't work. We'll have to go back and figure that out for the next time the virtual machine runs. How very exciting for me. But yeah, does anybody have any questions? The question cannot be, why didn't it run? We don't have an answer to that yet, okay? Any other question other than that is acceptable. Nope. Going once, going twice, everybody's burnt out. How delicious does a beer sound? YouTube, that's not for you. It's probably in the middle of the day. You should be working. Don't go have a beer right now. But that's it. That's today.